here we are. We're talking now about proteomes, and then we'll maybe come back and talk a little bit about atlases if we can. And so basically this is, you guys are kind of guinea pigs. This is now, our BioTools should work on a very good proteome comparison. I've heard that it's been finished now. We had various versions of it. The initial versions to compare proteins was taking way too long. And then I had a student that had a faster way, but it turns out there were some problems with it. It didn't really work all the time. And now some other people have written a really, really nice program, and Mike Lightsey has taken time to go through an enormous amount of codes and get it into this to work. And I've heard so far that it worked, but we'll keep in touch and see what's going on. Okay, so it's slowly kind of warming up a little bit outside, and we're here, proteome comparisons. So this time, I think there's only two questions. When are two proteins considered the same? Anyone want to answer that? I heard someone start to say something. What do you think? How would you define two proteins as the same protein?
If the primary structure, which is the amino acid sequence, is identical.
If it's 100% identical, it's the same. But what if it was only different? Let's say your protein is 300 amino acids long, and 299 of them are the same. There's one that's different. Is that the same protein?
No.
No.
Well, no, but it's pretty similar, isn't it?
You could call it the same if they have like the same protein-protein interactions and the same ligands and everything, couldn't you?
Yeah. So, I mean, absolutely, if it's 100% identical, that's easy. But in biology, very rarely do you do sometimes, but a lot of times you have things that are similar. Okay, so this is a problem that's been around for a while, and we'll come back to that. And then so now what you want to look at is you want to look at a bunch of proteins, and you have several hundred bacterial genomes. How are you going to do that? Talk about protein-protein interactions. You could do that, but that'd take a bit of time, wouldn't it?
Protein alignments, maybe?
You could do alignments. You could do cluster. And for several hundred genomes, it wouldn't take that long. Might take a couple days. Now, do you remember from earlier how many genomes are in Genbank? Anybody remember? Silence. Come on. Andrew, how many genomes are in Genbank? How many? We asked this question earlier.
60,124. Oh, that's 60,124.
No. Well, that's the ones that are finished, complete. But how many total are there, including the draft? Do you remember that? Because a lot of times the draft has most of the proteins, and in fact, something like almost 90%, 80 or 90% of the draft are good enough to work with. Some of them are not, but most of them are. What's the total number? If you go to that page and you say, how many prokaryotic genomes are there? What number do you get?
Well, for prokaryotic, there says 318,532.
318,000, right?
Yes.
Well, this question about just 100, now you're looking at 300,000. So we just happen to have on, I think, Visnu, are you still there? Visnu, are you?
Yes, I'm here.
So we just happen to have an expert here, Dr. Wankai, finished his thesis last autumn. And so Dr. Wankai, can you tell me how long it would take to compare 300,000 bacterial genomes, all the proteins in them?
16 seconds.
Ooh, well, that's using your method, but how, if you were to use
Oh,
cluster,
yeah. So yeah, generally, if
Yeah.
you want to use the compare sequence similarity, right, you need to compare one by one, right, all against all. So in the total proteins that from 100,000 or 300,000 is around billion proteins, right? Compare billion proteins
a
probably
billion
take
proteins
you,
to compare.
yeah, on a million machine, a million cores machine will take you 100,000 years.
So if you had a million
Yeah.
cores, it would take 100,000 years. So yes, you could do that, but it's going to take a bit of time. And you can be assured by the end of the 100,000 years, they would have sequenced a lot more by then, right? So yeah, and then once you've done all the comparisons, you have to somehow visualize that, right? Something to think about. So the last question is, so sometimes, as you saw, we have some genomes that are crap, right? So how can you, what's a good way to compare for bad assembly for the same species? And again, something to think about. I'm not gonna, we could, that's what the whole lecture is about. So, okay, so we start off, I kind of had already talked about this before. Back in 1997, the first bacterial genome was sequenced by Fred Blatter. Actually, I'm sorry, this is the first E. coli genome to be sequenced. The first bacterial genome sequence was in 95, 2 years earlier. E. coli is, I think, the 10th, something like that, bacterial genome to be sequenced. They'd started on it earlier, but they just kind of didn't get that together. But okay, so E. coli genome was sequenced 97. And so you could play around with it, you could look at it, and one of the things we could do is, of course, we built an atlas for it. And this is, the atlas was developed around these. And so the idea, so we first published this in 1999. And the idea is, here's the E. coli genome, and you can look along, the genome is 4.5, almost 5 million bases long. And as you go along, you can look at the AT content in the inner circle here, and then we're looking at the GC skew. So these are, it's more, which the G's are biased towards the leading or lagging strand. So here you see lots of G's, and here you see lots of C's. And so here's your replication origin, and your terminus is over here. So this is the leading strand going up this way. You can look at repeats, and these are various parts of the genome that are repeated. And here's all the genes, and then here's structural properties. And that's what that lecture that I just skipped, we'll talk more about this later, but that's an atlas, and you guys can make atlases for it. That it works for one genome if you have one finished genome. But again, if you have 300,000, you're not going to make 300,000 atlases. But sometimes it's useful for looking at structural properties. So, we have, at the time in 97 and 98, for a brief period of time, I said we have the E. coli genome. And it is, the E. coli genome is 4,693,021 bases long, and it has 4,328 genes. We know that. This is E. coli. Well, that was in 97 when that was published. 2001, so 4 years later, they published another E. coli genome. And the interesting thing with this one is it's the same species, Escherichia coli, but it has 1,387 new genes, which was really surprising. And so what you could do is you could compare, here's the first E. coli, and this was a common laboratory strain called K12, and this is isolate MG6055, and this is the first one that was sequenced, and it has 4,267 genes. Now, the second E. coli they sequenced has 5,499 genes. And what you could do is most of those genes are going to be, they're overlapped, they're going to be the same. And so you have what's called a core. So this is the set of genes that are shared between both of these genomes. And you'd expect most of them because these are both E. coli after all. But so you have 1,387 that are specific for that O157. And you have 693 genes that are unique for the K12. So the core is the intersection, and the pan is the total of all of these together. So the pan would be 3574 plus 1387 plus 693. That's the pan genome. Okay, but so I say that these genes are the same, but they're not always exactly amino acids. They're close. But so how do you really know when two proteins are the same? And again, this is in the 90s, and basically Eugene Koonin kind of came up with what's called the 50-50 rule. And if you take two proteins, you line them up. So you have one protein here, you're comparing it to this protein, you're comparing these two to each other. And you have a region that overlaps between them, okay, alignment region. If at least 50% of the length of your query overlaps with the subject, right? So if at least 50% of it overlaps and that 50% is within that 50% is also identical, what you're going to get is it's called 50-50 rule. Then you say these proteins are the same family, not identical protein, not the same protein, but the same family. Okay, so they're cousins, if you will, right? But if you think about it, it could be the bare minimum is you only are at 50%. It has to be at least 50%, right? But let's say you're exactly 50%. And within that 50%, it's only 50% identical. That means only 25% of the amino acids are the same. So this is a fairly lax rule. When we do these comparisons, and sometimes people say, "Oh, but what if you played with it? What if you made it more relaxed?" It's like, well, 25% identity is pretty relaxed as it is. Using this criteria, not a single protein is conserved across bacteria. Not one. No protein, none. Name a protein, it's not conserved. And a lot of people don't believe this. Oh, but we know these proteins are really important because no, they're not. And at the level of amino acid, at this level, 50-50, so at 25% identity, they're not conserved. And this kind of goes against what a lot of people have in mind. Again, this goes back to what I was talking earlier about in the 1950s and 1960s from computer science, they think of information science, combined with the idea that evolution is very slow and that changes aren't there. And that what's actually the information is, so you have this amino acid sequence and this makes this protein and this hardly varies. And this was an assumption that people had early on, but it turns out when we start looking at more and more proteins, guess what? That ain't true. It's not conserved. And so yes, you have ribosomes. Yes, you have things that are kind of ribosomes, but they're not that conserved. What you need is certain amino acid sequences to be in the right place, but there's a lot of different ways you could do that. So 50%. So I probably later on you'll see some stuff and you'll say, really? That's all that's conserved? Well, yeah. And so diversity is really a lot greater than people think. Okay, so that's just so you know, that's the criteria, 50-50. It's called 50-50 rule. And so here is one of these. This is from Lars' paper, 2004. So this is, what was that, 17 years ago. One of these papers. And what we did is we had a set of 1, 2, 3, 4, 5, 6, 7 genomes compared to each other, Pseudomonas genomes. And if you compare a genome to itself, of course, it's going to be 100% identical. But then what we say is, okay, what kind of duplicate proteins are there? In other words, if you throw away the perfect to itself, right? What other, and sometimes you have homologs. And so you've got, 0.1, 1 or 2% homologs, but not that much. So that's your diagonal. Now, if you compare this Pseudomonas to this Pseudomonas, you're going to find 94% of the proteins you're going to find homologs for. And so these are both originosa. Now, this is a Putida, which is a different species. So you only find 63%. And so this is kind of a way of looking at the proteins, comparing them. And so this is called a matrix. We call this a blast matrix. And again, here now we say the alignment needs to be at least 80% and the E-value needs to be at least 10 to minus 5. So you have some criteria for this. So, and this is from a review article 10 years. And so now we're comparing some Vibrio genomes. And these are Vibrio vulnificus. These are the same species, and then these are different species. And even, so vulnificus compared to another vulnificus. So let's see, that would be down here, they're 80%. That's, 84% identical to each other. 84% of the proteins have homologs. And so, again, now these are all E. coli, and this is E. coli, 2 different K12s, and they're going to be 96, 97% identical. So that homologs, 97% of them are homologs. What we could do is we could also color code this now so that you could see, and if they're strongly similar, they're more green up here. And then this within the genome, this diagonal, we can say if you have lots of duplicates, it's more red. So what this means is out of 4,126 genes shared between these two, 564 are out of 4,126 in this particular genome, in this strain, there were 564 homologs internally. So that's 13% of the proteins have internal homologs. Basically kind of duplicate genes, if you will. So the diagonal is looking at internal homologs, and the rest is looking at the homology between them. Now, if in fact what we do right now, this is a little bit different depending on so the matrix is not symmetrical because we compare this guy to this guy, and it's going to be a little bit different going the other way around because you look at how many genes there are, and there's a different number of genes. But instead, what we could do is we could look at gene families, and now it would be symmetrical. So now we have this small matrix here. We have 4126 proteins, but they represent 3797 families. And remember, we're talking about families now, if we're kind of defining it with this 50-50 rule. So you have 3797 families. This guy has 33,912 families. Combined together, they have 4,027 unique families, of which 3,685 are in both. So it's 90%. So now this is, you don't need to do, because it's going to be symmetrical because you're comparing both ways. So you only do half the matrix, if you will. And so this is a way of looking. Now, these are all E. coli. They're 90% identical. And then you have some internal homologs, which are down here. Now, we can look at these are all K12s, and they're all 90% or so. Now, we can look at another coli. This is an 0157. And do you remember the first slide I started? We had a coli, and now this is the 0157. And you see that it's actually only 61% in terms of these homologs here. And so you'd expect, because these are all the same K12, so they're going to be 90, they are 90% identical. But this is only 61%, 62% identical. And so, you know, this is back in 2006, we compared 70 coli genomes, and here's a little matrix, and you can see how similar they are. Now, the interesting thing is, as we started beginning to compare more, we realized that there's quite a bit of divergence, even within E. coli. So here, just to put this in perspective, if we're looking at humans versus gorillas, chickens, worms, yeast. So here's a human, Visnu, poor Visnu picking on him again. And the humans, so we say humans have 23,621 genes, gorillas have 19,000, and of those, 19,568 are orthologs. So it's 99% identical to humans. You probably heard this before. Chickens, on the other hand, have 14,000 orthologs. So they're 76% identical. Worm has 10,000 orthologs. They're 50% identical. And yeast is got only 28%. And you guys probably know this, right? This kind of makes sense because, you know, gorillas, humans, 99%. Chickens, 76%. That's fine. But now these are all the same species, E. coli, E. coli, E. coli, E. coli. Now it's Yersinia, but so this E. coli has 4,150 genes. Now, when we compare another, this is the same strain, K12, but a different isolate. It only has 3,843 that are the same between these, so it's 95% of the orthologs are shared. This is a commensal strain. It's only 76%. Oops. Oh, come on. Come on. Sorry. My mouse is sometimes sensitive. So this is 76%. This is 60%. By the time that you get down to 29%, now it's Yersinia. Now you're kind of outside the coli family, but you're still in a gamma protobacteria. You're still in the kind of in the coli, friends of E. coli. So the point is that you see this enormous diversity within these genomes. So now there's a lot of caveats to this because these are all macroscopic. These are really big things, and there's a lot of selective pressure going on. These are all multicellular organisms. But there's an amazing amount of diversity here. So this is a friend of mine adapted it. This is a Spanish book, and he kind of has showing the same kind of comparison between this is looking at the pan genome. So you've got that. We'll talk more about that. But, so what can we did on these genomes, and this is several years ago, but what can high throughput genomics teach us about evolution? So they asked me to write a review article about Fred Blattner had published a paper where they had used synthetic biology to create a new E. coli genome, to reduce E. coli. And so I started off by saying, well, why would you want to design a better bacterium? And so, you know, if you really understand how something works, you can make predictions. And so synthetic biology is all about understanding life by constructing it. If you really understand it, you should be able to engineer it. And so there's a lot of progress and been going on. But, you know, the interesting thing is there's this paper by Science, which they had asked me to review, and they basically use synthetic biology to reduce E. coli. And they reduced E. coli about 5%. They're very proud. You know, they got rid of some of the genes using synthetic biology, which is good. But my question is, so what about evolution? And what, when we start looking at E. coli and friends, remember there's an enormous amount of diversity within E. coli in terms of size and how many genes there are and things. But then if you start looking, so these are all E. coli's, the blues are E. coli's, and you see there's 4,300 genes here, 5,300 genes here. A Shigella is essentially E. coli, and this Shigella has 4,073 genes. And you have these other, remember we're talking about these endosymbionts down here. These guys are all essentially cousins of E. coli. And so you look at this guy, Carcinella rudi, it only has 182 proteins. And in fact, so here's the coli that they reduced had 5,066 genes, proteins. This has 182 proteins. So this is only 3%. So in fact, real biology, and by real biology, I'm talking now about evolution that we can watch and we can look at, can really teach us a lot in terms of being able to look at what's going on in the world. Okay, so this is from a paper back in 2005. So this is 16 years ago, yeah. And this paper, when it came out, Tedlin, this is basically looking at, this is kind of defines what is a core and pan genome. And so this paper was very much kind of setting the standard. And what happened is they looked at that time they had 8 Streptococcus genomes, and that was a lot back then. This is 16 years ago. And so what they did is they took all 8, and some of them are bigger. So here's how many genes there are. Some of them, so on average, you have around 2,200 genes. Some have 2,400, some have 2,000. And what they did is here's all 8. And they said, we take these 8 genomes and we average them, and this is how many genes we get from those 8, okay? And so then you start with that. Now you say, okay, now you start comparing. So you have 8 of these, and you say, okay, you look at how many genomes. So now you got a second genome. So you start with this genome up here, and you add the other 7. And you start with this genome, you add the other 7. This genome, you add the other 7. So you have 8 times 7 possibilities there. And you take these and you multiply it by the remaining 6 possibilities times the remaining 5 times 4 times 3 times 2. And then at the very end, you have, now you've compared 7 of the 8. So you just compare it to the last genome, and you compare it to 1, right? And you do the average there. So you have what's called 8 factorial. So that's 8 times 7 times 6 times 5 times 4 times 3 times 2 times 1. And for 8 genomes, you could do it. It's a bit computationally expensive, but you could do it. And so they could find the core. And so they found around 1,800 genes in the core. And then similarly, they're looking at new genes. And I love this figure because this is done by, this guy, he's a mathematician, a French mathematician. He's very smart. But the reason I love this is what this plot is showing is that as you sequence more genomes, on average, you're going to get 30 new genes every time you sequence another genome. And this is going to go on forever, for infinity. So if you sequence an infinite number of genomes, you'll have 30 times infinity. It's like, well, this doesn't sound right. So there's some problems with that. But that's kind of, anyway, it's very mathematical. And so what you do is, here's the number of new genes added. And what you could do is the core is dropping, right? But the total number of gene families is going to go up, right? And so this is what's called the pan. So the core is dropping and the pan's going up. Now this paper, one of the interesting things in this paper is they actually say that they did 8 genomes. Again, this is 16 years ago. But they said, surprisingly, they found 8 genomes is not enough to identify all the genes. And mathematical modeling made the surprising prediction that even hundreds of genomes might not be sufficient. Okay, this is 2005. So I showed you this plot earlier. So this is when the Untitled paper came out. And they say, oh, you might need hundreds of genomes. Well, guess what? we're over here. And so now when you start looking, you have 20,000 E. coli genomes. And so you can start doing lots of fun comparisons. So, originally I was comparing, we had 1 E. coli, then we had 2, and then 4, and then, and now it's just, so here's 7 E. coli genomes, doing some comparisons. And then Henning started looking at 32, and she tried to use Tedlin's method, but it was just, it didn't work very well. So what she wound up doing is a heat map, kind of doing all these comparisons, looking at, so here you can kind of get the core, doing a variety of extrapolation methods. We wound up with around 1,500 core genes. And so this is Hattie. So she was a PhD student working on this. And if you look the way that Tedlin did it, you have 32 factorial. So that's 2.6, 10 to the 35th possible permutations. That's a very, very big number. So you can't, that's not really going to be that helpful. So what we can do is, so this is looking at 30 genomes. And what we've done is we basically, over time, we add more genomes. So the core goes down and the pan goes up. As you sequence more genomes, so you're going to get the total number of possible gene families goes up. And so here's all the genomes. We have 30 genomes, and you can see the core goes up and the pan goes down. Okay, and there's a line that's the core, and then I'm not sure. Oh, that's just the reference. Okay, so we wound up with around 1,500 core genes, roughly. Okay, out of E. coli. Now, typically E. coli has around 3,000 genes. We're saying on average, well, there's people have known for a long time, there's around, but a typical E. coli genome, remember, has 5,000 genes, but there's roughly 3,000 kind of well-known, well-characterized E. coli gene families. This is a little surprising. This is only half, but we thought, okay, and that's with 30 genomes. Now, a couple of years later, another student, this was Oksana, the core now we're doing 64 E. coli genomes. We went from 30 to 64. The core now drops to 960, which is getting kind of small. We get a pan of 15,000. And if you look here, this 63 factorial, that's 2 times 10 to the 87. That's a very, very large number. And so this is Oksana's work. And this is published 2010. And then Rolf comes along, this is 2 years later. Now we have 187. So now we start comparing 187 genomes, and we wind up with a core of around 3,000 and a pan of 23,000. Now, how do we get this? Because it was going down. Remember with Oksana, it was below 1,000. We realized things were going on. What we did is we did something called a slack of 19. And what that means is that the core needs to be present at least 90% of the genomes. And so if we have 187 genomes, then it needs to be present in 18.7 or 19 of the 187 genomes in order for it to be considered. And by doing that, we throw out, because it turns out that sometimes you get a bit of schlop. And so you need to kind of throw out sometimes bad genes. 187 factorial is way too big. So this is part of Rolf's PhD project. And so this is 187 E. coli. This is 2012. Now, 2015, 3 years later, now we're comparing 2,000 E. coli genomes. And this is with Miriam and Seyron. And now, again, the core is staying around 2,000 or 2,085 coli genomes. The core is stable around 3,000 proteins. And the pan is already 90,000. This is 2015. And then, so this paper, and it's actually, this is out now, it's published. I'll give you the reference. I should include that with this. It came out actually just, I think in January, February, in Nature Communications Biology. So this is Zulema and Caleb, a PhD student. This is for 10,000 E. coli genomes, but we actually, these are the ones that were complete, but we actually have 100,000 E. coli genomes that are in the SRA. So we compared more than 100,000 E. coli genomes looking at the Panacore. And what we're looking at here is, so this kind of this greenish color here is the core. There's around 2,800 that's found in all E. coli. And then we have the phenotype A, and these guys are the core that's in phenotype A that's not found in the others. And then this is found in phenotype B1 that's not in the others. And so, and you can see that the Shigellas, you have lots of extra genes. In the L157, in fact, you're going to have, if you look, this is almost 4,000 genes that are unique to the O157 that aren't found in the others. And so this is kind of a way of comparing and visualizing, comparing across 100,000 E. coli genomes. And so we can look at the lineages and some of the things that are going across. We can do heat maps. And so Caleb actually has, so this is this is just looking at 25 E. coli so that you can see the heat maps comparing each other. So these are the K12s and these are the 157s. And basically these K12s here are pretty similar to each other. And the 157s are pretty similar to each other. That's what this darker color here, this really dark turquoise, means they're very similar. The diagonal, they should be because it's the same genome, right? But then you can see some others that are very similar to each other. And the brown means they're not similar, they're very different from each other. And so this is, I showed you 25, now this is 10,000. And he has a similar heat map for 100,000. And so the point is that this is a way of visualizing now. You're looking at basically comparisons across 100,000 genomes, going from originally, remember we were comparing just 2. And so Lars is a good friend of mine, collaborator. Well, we have a student, Christopher, that Lars and I spent a lot of time on the whiteboard with Christopher Keel. Christopher is more kind of mathematical. And we're looking at Panacore, and the K12 genome, this one has 4,144 proteins. If we look across lots of E. coli, even back then we knew there was around 100,000 gene families. And there's only around 3,000 core that's found in all E. coli. So a typical E. coli genome actually would have roughly 5,000 proteins, 3,000 of which is in the core in all E. coli, but there's 100,000 gene families. And this is published back in 12 years ago. So we could do these heat maps, and now this is 28 E. coli genomes, and these are all the K12s and kind of these are the B type. And so we could look at different things within those 28. As you can imagine now, these are 61 E. coli genomes, and then this is 130. Whoops, this is 130 E. coli. And so it starts getting kind of big, starts scaling out. Okay, so that's just kind of, you're going to make some of these, but a dozen genomes is fine just to get the idea. So now this is kind of an interesting thing, because what we've done is we've looked at this particular genome, and what we've done is looked over time. So this is 1998, this is 2004, 2008, 2009, and then these are different models for annotation. And what you see is that it turns out that this model, there's the J. Craig Venter Institute that they had, they're predicting way too many proteins that are not shared with other things. And so, but this is the same genome, the same sequence, which is kind of fun. So it depends on which gene finder you use. And that's one of the reasons why in your computer exercise, you download a set of genomes, you run the same gene finder through it. So now you've got consistency. And now if you're to look at the same genome, you should get the same thing. Just as this is kind of as a cautionary tale, because you can see a lot of differences here just looking at different versions of the same genome, because they're annotated by different people with different cutoffs. But quite a bit of variability there across that. So Pseudomonas, and this is a wonderful paper from Assemblies, one of my students. And the question is, Azotobacter is supposed to be a completely different genus outside Pseudomonas, but if you actually look where Azotobacter lives, See, this is all green here, and see, these are all Pseudomonas. It should be Pseudomonas. There's lots of evidence it should be. But you start looking at these other guys, and you see compared to all these other cousins and friends and things, that this is a Acinetobacter, Saccobacter, some of the other things that are in the same family. Azotobacter should be in the Pseudomonas family. And again, this is using this 50-50 rule we're talking about earlier. So, and this is Vionella. Vionella is another interesting protein. If you guys, another interesting genome, a species, genus, if people are interested in this, looking, comparing organisms. So Vionella is found, among other things, it's really abundant in your mouth. And it's also found in your lungs. Okay. And you say, well, yeah, all right, it's a bacteria. But it turns out this bacteria is an anaerobe. It dies in the presence of oxygen. So how in the world can it be abundant in your lungs and abundant in your teeth, in your mouth? Well, it turns out that it forms a biofilm with another bacteria, which is an anaerobe. So this other bacteria, lots of oxygen, but on the inside of the biofilm on your tooth, where there's no oxygen, is where Vionella lives. And Vionella is kind of a weird thing because it's not really gram-positive, gram-negative. So technically, it is a gram-positive bacteria, if you look at the way that it's kind of, it should be kind of, but it has two membranes, so it's gram-negative. So it's one of those weird things. It shares 2% of the genome, 2% of the proteins it shares with coli, 5% with Clostridium. So this is gram-negative, this is gram-positive. I mean, so this guy's just really, really different. It's similar to itself, but it's really very different than each other. This is Vionella probably diverged a very long time ago. So it's just kind of an interesting way of looking at this. This is looking at Salmonella, again, between organisms, different species of Salmonella. This is looking at a whole bunch of Clostridia genomes, 60 Clostridia. These are all Clostridia botulinum genomes. But these are also Clostridia botulinum, and so are these. And the reason for this is that botulinum, the way that this species is defined, is do you have this toxin? Well, the toxin can live on a plasmid. So you can have genomes that are very, very different, but if you have the plasmid, it's called Clostridia botulinum. So that's just kind of a little bit of an example there. So again, these are some E. coli genomes, again, comparing. This is what's called a blast atlas. And Vishnu, I don't think we can do blast atlases yet. Is that right? Vishnu,
That's correct. Yeah.
So we can't do blast atlases yet, but that's the last thing that we're working on. Hopefully we'll have that soon. But what the blast atlas is a way of comparing this E. coli to others. So what you're doing is you're blasting. You see these regions here, which are missing in the other genomes, but are present in the reference. We talked earlier about Methanopyrus candelaria. This is the archaea that lives at 110 degrees. And there's some gaps in there that are missing from other organisms. And these probably form some kind of capsule on it. This is from BMC, that's 2003. Geez, that's almost 20 years ago we published that. Hard to believe. So this is looking at Streptococcus pyogenes, yeah. So this is basically, again, just looking at regions that are missing. So here is Vibrio. This is from a Haiti outbreak, and there's a region here. There's basically this thing got inserted in the reference genome here, but also you see in one of these strains here. These are strains from Nepal. So this is from the outbreak in Haiti. And then the black is commensal, and these are looking at these are looking at different Vibrio cholerae. And generally, so you see this gap that's missing, and that's this particular, it's kind of an insertion sequence here. But you have other regions that are missing that are surface proteins. And we don't know what this one is, but this is a whole bunch of proteins that are involved in surface proteins. That's there. And so what happens is you have genome enrichment. You have the same kind of thing we talked about before, where you have things coming and going, making the genomes bigger, smaller. And so now this is one of Oxana's atlas. This is of comparing. There's 116 circles on here. We're comparing 3 billion comparisons, because what we're doing is we're comparing other genomes to this one reference, and this one reference, this strain contains around 5,000 proteins. So we're comparing 5,000 proteins to the other E. coli genomes, which have 5,000. And so in this one figure, you're looking at 3 billion comparisons. And so if you were to actually look at one of these blast reports, so you do an alignment and you look every second at one blast, it would take 92 years to look at this data. And the point is that this is a way of, again, of visualizing this so that you can zoom in and you can look at particular regions and you can say, look, there's these gaps here. And there's these dark regions. These are things that are highly conserved in these different strains. And so that's, sorry, my mouse, a very sensitive mouse. So the point is that this is a way of visualizing massive amounts of data, being able to handle big data. And that is the end of this. So these are looking at lots of bees coming around. So when are two proteins the same? When are they the same family? Now, see, now I define myself. Do you want to try again, Andrew? Anyone?
I'm here. I'm just thinking.
The same family will be when 50% of the sequence length has a sequence, a 50% identity.
Yep. So if you think about that last slide I showed you, how do you compare similar protein coding genes across 100 bacterial genomes? That last slide I showed you, there was 118 genomes. How did I do that?
Using the biotools?
You want to elaborate a little bit? How did I do the comparison? I showed you a way of doing it, and I talked about if you're to actually look at the reports themselves, if you're to look at the blast report for each protein comparison, right, it would take you 92 years. Do you remember that?
Yes.
But obviously it didn't take 92 years to look at that figure. So what did I do? I actually showed you those results in one figure. So what's going on? How did I do that? So what I did is I have a reference genome, I have a little circle, and each of the different rings represents another genome. And in that ring, I say, compared to your reference, so you have your protein in your reference, compared to that reference protein, what's the best match I can find in that particular genome for that circle in the ring? And if there's nothing there, it gets white. If there's a really strong match, it's darkly colored. Okay? So it's a visualization basically of the maps, but now you're mapped onto space of the chromosome. So you can see regions of conservation and regions where things are missing, right? So you look along the chromosome and you can see what's called genomic islands. So it's now the caveat is this is compared to that reference genome, but you can still take that reference genome and compare it to 100 other genomes. And so that's going to represent 1 billion different protein comparisons. Now, in Visnu's case, now instead of 100 genomes, we're comparing 100,000 genomes. Now you have 1 billion proteins to compare. So now it's 1 billion times 1 billion. And that's where Visnu said, if you had, what was it, Visnu? Was it 100? Was it a million core machine?
Yeah, this is the million core machine.
So if you have a computer with 1 million cores, it would take 100,000 years to calculate. So that's why you need a better method. And Visnu, sometime he can tell you about his method. I'm sure Visnu would be happy to tell you if you're to ask about how to do that. He has a paper published on that. Okay, I didn't really talk as much about this, but basically, sometimes you have bad assemblies, and one of the things you can see when you're doing your blast matrix is you have a genome that just, so let's say you have a bunch of E. coli genomes, they're all 96% identical, and one of them is only 3% identical. We say, well, wait a minute, that's something's going on there. So that's one way of looking at quickly checking whether assembly is bad or not. And sometimes when you pan core plot, what happens is you're going along and you add a genome that's crap, and also the core drops and the pan jumps. And then you know, okay, so there's something screwy going on there. So that's the end of that.